Hello everyone, Lauren Bean here. This is a video for the sheep brain dissection. So what I have in front of me here is a nice clean brain. I have a scalpel blade and a wooden probe. And that's really all you're gonna need for this activity. It would be a good idea to have the dissection map provided by the Carolina Distance Learning Lab Kit. That's a great guide for you. But I am going to go through and identify and talk about the main features and structures that I want you to be able to recognize. It's a great idea to be looking at the diagrams from the central nervous system chapter, looking at those brain images, and you want to be noticing a lot of these correlations. So right under the lab folder, you will see a nervous tissue and brain lab document, and there's some great visuals there for you. I'd recommend having those out as well. You're going to be going back and forth quite a lot. You may feel like you have to take a glove off so you can flip a page. You do whatever you got to do. Make the most out of this dissection. All right, so let's start off by talking about some general features here. We want to position and orient ourselves first of all. So here is the front of the brain, the anterior region, or the rostral region. And rostral means toward the forehead, while toward the back here, posterior or caudal, that's meaning toward the spinal cord. With a sheep brain, you're going to notice that it's kind of flattened and compressed. It's more elongated and therefore like an animal. So the brain and the spinal cord, the brainstem, it is positioned a little bit differently than ours. We are bipedal, we stand upright. So we have more of a spherical brain, and the spinal cord is angular downward. But what you're going to see is that all of the features are the same, right? So mammals, their brain structures, very, very similar. The biggest change amongst the human species is the increase in the cerebrum, right? Cephalization, that thinking brain getting larger, making us unique individuals, unique species. All right. So I first want to point out that we have a very distinct left and right cerebral hemisphere. So as a whole, this is the cerebrum. Then we have our little brain back here, the cerebellum. So notice that the cerebellum and the cerebrum, they're quite separate from one another. You can lift it up and really notice like how they are isolated structures. They are connected due to the brainstem, but you'll see there is this transverse cerebral fissure here causing that separation, or transverse cerebellar fissure. Dividing the left and right cerebral hemispheres is this deep median longitudinal fissure. So you can pick your brain up, you can kind of use your hands and separate those two halves, and you will realize there's quite a narrow cavity there. You can even use your wooden probe if you need to separate the tissue a little bit to see in there a little better. You could use your scalpel blade at this point, but you don't want to get too carried away. The best tool during a dissection is your own hands. Okay. Toward the caudal end here, extending out, we're seeing the spinal cord. We do have the brainstem as well, but we can't see a whole lot of it with this view, so I will be flipping the brain over. But you do have your different lobes. So your frontal lobes toward the front, parietal lobes, temporal on either side, coming back toward the occipital lobes. One other thing that we can identify here are the sulci and the gyri. So each ridge being a sulcus, then these shallow little grooves here being a gyrus, collectively called gyri, or plural would be gyri. Then if we flip the brain over, of an inferior view here, we can see a lot of other structures. Some parts are, not really left intact. It's really going to depend on your sheep brain, so you may see some variability here from what I'm showing you in this video, and that's perfectly fine. So main regions I want to identify first, so we can see components of the cerebrum still. Cerebellum is kind of covered by the brainstem at this point. So for the brainstem features here, we have the midbrain. Kind of see a left and right component to that. Then we have this the pons. Okay, that component right there. then the fat kind of medulla oblongata there, leading to the narrow spinal cord. So midbrain, pons, medulla oblongata, leading to the spinal cord. What we can also see are some nerves, some tracts, starting at the top here, which my brain in particular doesn't have a lot of this tissue remaining, but what you may see are these flappy feet that we call the olfactory bulbs. I have a little bit of tissue left behind there of that, but these are swellings of the olfactory tract, and the bulbs lay in the superior region of the nasal cavity. Olfactory neurons dangle down from it and are responsible for our sense of smell. So olfaction, olfactory has all to do with the sense of smell. So that bulb then leads to this olfactory tract being shown there. So this side I can see it okay. This other side I really can't see it much at all. It's going to depend on the brain that you have. Then coming down here, we see this white tissue creating a V-like assembly. So what we have is the olfactory tract on either side, left and right olfactory tract, crossing over at what we call the optic chiasma right here. And then what we would have is an extension of left and right olfactory nerves. So unfortunately with my brain, the nerves are not left behind. They were removed as, this organ was pulled out of the skull. But again, you would normally see an extension of this white tissue, the left and right olfactory nerves, and make a perfect X. So you may find it useful to look at your dissection map for that. Not a great visual of it. Make sure you do look at the diagrams I have posted on the Brain Lab document because you'll get a better understanding of that assembly. Then next to that, we have the pituitary gland. So fortunate for us, this was left intact. So here's that gland right there. Actually has an anterior and posterior component to it, so it is bilobed. This is the infundibulum, the stalk from which the pituitary gland hangs from. And if we were to poke a hole into that infundibulum, it would travel into the hypothalamus. Collectively, the hypothalamus and pituitary gland have all to do with making hormones for the body and have all to do with regulating our endocrine system, our hormone system. So very, very important brain structure there. Then we can see more components here of our midbrain, a couple different structures associated with that. We have the cerebral peduncles, kind of left and right, these distinct features right there, creating really a connection point of the midbrain to the cerebrum, really tying together the brainstem to our larger thinking brain. One way to examine cool features of the midbrain is to take your sheep brain, flip it over in our starting position, our rostral and caudal, anterior, posterior. I want you to take your thumb and put it on the cerebellum like this, your other hand toward the front of the cerebrum, and you're going to kind of fold this structure down like this. So I've even stuck my thumb in here even further into the midbrain, and I'm causing some separation here of our brainstem, our cerebrum, and the cerebellum. You want to be careful. You don't want to just completely rip it off, but you need to be aggressive enough that you are seeing the separation and you get to see these cool features on the inside. So here I have the cerebrum, our left and right cerebral hemisphere. I'm seeing features of the midbrain here, and then you're going to see this cool little structure called the pineal gland. It is wrapped in those different meningeal layers, and you see these darker strands. Those are all of the different blood vessels. So you may need to separate it a little bit, but here we see the pineal gland. It's all to do with producing melatonin, regulating our day and night cycle. Then you see these 4 distinct bumps. This is called the corpus quadrigemini collectively. We have a pair of superior colliculi, which have all to do with visual tracking, visual reflexes, and then these smaller ones are the inferior colliculi, which have to do with auditory tracking. So that automatic response we have when we see something out of the corner of our eye, we turn toward it, or we hear a noise and we kind of turn toward it and we try to figure out where that is coming from, right? That automatic reflex that we have controlled by these brainstem structures here. Peeled back the cerebellum, so you get to really see just how separate it is from the cerebrum. Any communication going to and from has to pass through the brainstem, pass through the midbrain and the pons to get to that point. All right, so now we are ready to cut our sheep brain so we can have a nice internal view. We want to create a medial section. So we're going to cut right down this median longitudinal fissure here. It's a great guide for us. Nice clean cut all the way through that entire brain. I'm just cutting. The brain is quite soft, so you should have no issue cutting through. Nice and sharp scalpel blade, so that's pretty great. Then you just kind of fold it open like this. You'll have a nice mirror image of the different structures. All right, so I'm going to use my right half here to go through the features inside of the brain. So we're going to notice that we have the cerebrum here. We can see the gyri and the sulci, and that's because there's quite a separation between the left and right cerebral hemisphere, right? And that's due to that deep longitudinal fissure. The point in which these hemispheres make contact is at the corpus callosum. So you're going to see a change in tissue types, a little bit smoother, whiter in color. This is where those fibers communicate between the left and right half. Just beneath that, we're going to see the opening into the right lateral ventricle. And as I'm expanding this ventricle here, what I'm noticing is that there's some septum pellucidum left there. That's a thin membrane that separates the left and right half. And when I cut down the longitudinal fissure, I kind of favored the left side a little bit. So more of the right side of the brain was left intact. So I'm going to use my probe and actually just separate that septum pellucidum, that thin membrane. And now I'm seeing into that lateral ventricle. It's quite a large structure. It's a fluid-filled chamber. Right, it contains that cerebral spinal fluid, and it's pretty decent size. So I just penetrated through that septum pellucidum into the right lateral ventricle. So fluid enters that lateral ventricle, and then it's going to meander its way down into the third ventricle. And really the third ventricle is essentially the hypothalamus. Here we have our thalamus, the egg-shaped structure, and then the hypothalamus is just below that, kind of funnel shaped. And again, the fluid moves from this lateral ventricle down the interventricular foramen into our hypothalamus. From there, that fluid then goes down the cerebral aqueduct into the fourth ventricle. So you can actually, if I fold it down a little bit more, we're seeing part of that cerebral aqueduct right here, which it travels down through. I left the pineal gland intact, so that's right there. Again, here we have the thalamus. Then we're getting down to the midbrain structures here, our corpora quadrigemini, so we can see components of that, superior colliculi, inferior colliculi, or you can even kind of flip it like this to help orient yourself as well, because here we have the pons, right? That's the area of the pons, and then the medulla oblongata there. Notice that it's closely associated with the cerebellum. So communication, cerebellum, cerebrum, spinal cord is passing through the pons, mostly and connecting to the brainstem at that point. With the cerebellum, what you'll notice is kind of this elaborate folded network within. And if you look really closely, the arrangement of gray and white matter makes it look tree-like. So we call that arbor vitae, that arrangement of gray and white matter. So pretty cool. You can make some additional cuts to take a closer look at that, expand the tissue, right? It's a very cool arrangement of neurons within there, right? So make the most out of this dissection for yourself. Look around, look at your dissection mat or that labeled piece of paper that came along with your distance learning lab kit. And I hope that it was a good dissection for you to understand the anatomy of the brain.